एक्सक्रीटरी सिस्टम ऑफ ह्यूमन। ये जो टॉपिक है ओनली 10th के लिए ही नहीं है, 12th के बच्चे भी इसे कर सकते हैं, उसी स्टैंडर्ड से इसके नोट्स क्रिएट करे हैं। एक्सक्रीटरी सिस्टम ऑफ ह्यूमन, different types of waste materials are produced in the metabolic activities going on in the body, which are not only unnecessary but also harmful, as CO2, water, nitrogenous waste, अमोनिया, यूरिया, यूरिक एसिड्स। तो हमारी बॉडी के अंदर कार्बन डाइऑक्साइड और नाइट्रोजनस वेस्टेज जो होते हैं, वाटर होता है, एक्स्ट्रा वाटर जो होता है, उसको जब हम डिफरेंट टाइप्स से उस वेस्ट को जो बाहर निकालते हैं, तो ये जो बाहर निकालने की जो एक्टिविटी है वो हमारा क्या है, आपकी एक्सक्रिएशन है। एक्सक्रिएशन क्या है, इसे हम डिफाइन करें तो द प्रोसेस टू एक्सक्रिएट आउट द नाइट्रोजिनस वेस्ट फ्रॉम बॉडी इज कॉल्ड एक्सक्रिएशन। तो जो भी नाइट्रोजिनस वेस्ट होते हैं उन्हें हम अपनी बॉडी से अलग करते हैं, इसको हम एक्सक्रिएशन कहते हैं। ओके, आगे हम बढ़ते हैं। एक्सक्रिएटरी सिस्टम, द नाइट्रोजिनस वेस्ट मटेरियल्स प्रोड्यूस्ड ड्यू टू प्रोटीन मेटाबॉलिज्म आर एक्सक्रिएटेड आफ्टर complex chemical reactions by specific excretory organs, okay, that organs of the body which help in excreting out the wastes are called excretory organs. Elimination of nitrogenous waste. Ammonia. Ammonia is produced due to decomposition of amino acids and proteins, which is very harmful for body. Those animals in which nitrogenous wastes are excreted in the form of ammonia are called aminotelic animals. Example: protozoa, perifera, hydra. Urea. Number second, urea. Urea is less harmful than ammonia. Urea is water soluble. Such animals which excrete mainly urea are known as ureotelic. Such animals which excrete mainly urea are known as ureotelic animals. Example: annelids, bonyfish, amphibians, rabbits, humans. Number third, uric acids. Uric acids is less poisonous and is nearly insoluble in water. Such animals which excrete mainly uric acid are called uricotelic animals. Example: insects, reptiles and mammals living in desert habitat. Human excretory system. See in diagram. Number 1, kidney. Number 2, ureter. Number 3, urinary bladder. Number 4, urinary passage or urethra. In human, one pair of kidneys are found, which are situated at dorsal side of abdominal cavity on both the side of vertebral column. The right kidney is somewhat anterior to the left kidney. A cap-like endocrine gland, adrenal glands, cover the upper end of the kidney. Number 2, ureters. A narrow tube with thick muscular ureter. A narrow tube with thick muscular walls comes out from the hilum of kidney is called ureter. Its proximal part in the kidney is wide and funnel shaped, which is called pelvis. Both the ureters starting from the pelvis open below. In urinary bladder. Number 3, urinary bladder. Its muscle bag like structure in which urine is collected permanently. There are three layers. Number one, external layer. Number second, internal layer. Number third, middle layer. External layer, serosa of peritoneum. Internal layer, mucus layer. Middle layer, unstripped muscle layer. Number 4, urethra. A duct comes out from the neck of urinary bladder, which is called urethra. Urine is extracted out through urethra. Human kidney, see the diagram. In human, each kidney is covered with firm fibers, connective tissues cover which is called renal capsule. Kidney is differentiated into two parts, cortex and number second, medulla. Cortex. It is situated inner to the capsule and due to malpighian. Cortex. It is situated inner to the capsule and due to malpighian bodies looks like granular. This is the outer region of dark red color. In cortex part, malpighian bodies proximal, convoluted parts and distal convoluted parts are found. Number second, medulla, interior part of kidney. In this Henle's loop and collecting tubules are found. There are seen conical elevation which are called pyramids. Pyramids is situated in a cap-shaped cavity which is called calyx. Calyx is joined together open into pelvis which is funnel shaped and open in urethra. Nephron structure, see the structure. Each tubule can be divided in the following parts. Number 1, malpighian body. The anterior part of each uriniferous tubule is called malpighian body, which is formed by two parts. Number 1, Bowman capsule. This is a cup-shaped structure in which the glomerulus is inserted. The wall of Bowman's capsule is thin and double layers. Number 1, epithelium layer. Number 2, specific type of cells layer called podocytes. Next part of malpighian body is glomerulus. It's found in the cavity of woman's capsule. Afferent arteriole from renal artery supplies blood to glomerulus, and afferent arteriole takes out the blood. Afferent arterioles divided into 50 branches. Okay. Number 2, neck. This part is a narrow small tube-like structure next to Bowman's capsule. Its wall is made of ciliated epithelium. Number 3, proximal convoluted tubule. This structure is a long, thick, coiled tubule next to neck. It is stratified by cuboidal epithelium, and on its inner edge many microvilli are found which together form the brush border. Due to presence of microvilli, the absorption surface is increased. Number 4, Henle's loop. It's the middle part of uriniferous tubule situated next to proximal tubule. Thin and U-shaped tubule part called Henle loop. It's differentiated into two arms, descending arm. Number 2, ascending arm. Number 5, distal convoluted tubule. The ascending arm of Henle's loop open in distal convoluted tubule, which is situated in the cortex region of kidney. Microvilli are not found on these cells. Number 6, collecting tubules. The distal convoluted tubule of uniferous tubule open in collecting tubule. Many collecting tubules together form a single thick main collecting tubule, which is called duct of Bellini. Formation of urine. Our next point, formation of urine. Urea formed in the liver cell is carried in the kidney. The blood with urea from liver is poured in the post caval by hepatic vein. Urea is separated from the blood from post caval. This is called urine formation. The process of urine formation is completed in the following steps. Number 1, ultrafiltration. In this point, Bowman capsule in the uriniferous sac functions as a microsieve. The afferent arteriole carries ureated blood in this, and efferent arteriole takes away blood from it. The wall of capillaries of glomerulus has innumerable pores of about 0.1 μm diameter. The substance dissolved in plasma can get through these pores. Blood corpuscles, blood protein, blood soluble colloids could not filter. The filtered plasma from the glomerulus is called glomerular filtrate. This fluid comes down in the cavity of Bowman's capsule. The process of filtration from the wall of blood capillaries of glomerulus of excretory and other utilizable substance is known as ultrafiltration. Number 2, selective reabsorption. The glomerulus filtrate produced by ultrafiltration contains urea, uric acid, creatinine, etc. Excretory substances also with glucose, amino acids, some fatty acids, vitamins, water and other useful salts. In the filtrate, 95% of the water in the blood also filters out. The process by which the useful substances are reabsorbed in the blood from uriniferous tubule is known as selective reabsorption. The process of reabsorption in the different parts of the uriniferous tubule is as follows. A. Reabsorption in proximal convoluted tubule. On the inner wall of this, microvilli are found. Due to this, reabsorption capacity increases 20 times. Complete absorb glucose, amino acids. Less absorb water, mineral salts. Least absorb urea, uric acid, creatinine, sulfate. Reabsorption in Henle loop. In descending arm, 35% of the filtrate absorbed by osmotically blood plasma absorb Na plus, K plus, Cl minus, less preable for urea, sodium and other ions. Next point C, reabsorption in distal convoluted tubule. The reabsorption of Na plus, Cl minus remains continued in this tubule. Here the filtrate is isotonic in comparison with blood. Here nearly 19% part is absorbed. D. Reabsorption in collecting tubule. Here water molecules are reabsorbed from the filtrate. Thus the filtrate becomes hypertonic. The wall of collecting tubule in its interior part is slightly permeable for water molecules. Number 3 point, last point, some harmful secretions. Some harmful materials like pigments, some medicines, uric acids, High puric acids etc. could not seep out at the time of ultrafiltration in glomerulus filtrate. These waste materials lacked free in the uriniferous tubules cavity by active transport. The by epithelial cells of Henle's loop and proximal and distal convoluted arms. This activity is known as tubular secretion. Okay, bye.